welcome everybody to today's Combustion webinar. Today's speaker is Johannes Kiefer from the University of Bremen in Germany. Johannes Kiefer is the Chair Professor of Engineering Thermodynamics and Dean of the Faculty of Production Engineering at the University of Bremen in Germany. He graduated with a degree in chemical engineering at the University of Erlangen-Nuremberg and a PhD from the same institution. He's been at the University of Bremen for several years now and has had a number of research stays at other organizations, the Sandia National Lab, Lund University, and elsewhere. So very well traveled and very wide breadth of research. And today, Johannes will be telling us about chemometric approaches for evaluating spectra from combustion environments. And so, yeah, I'll turn the floor over to you and yeah, welcome.
Yeah, thanks a lot, Isaac, and thanks for this nice introduction, and also thanks to the organizing committee for inviting me to give this talk. I think it's a great idea to organize this webinar in these kind of strange times where we can't travel to conferences and stuff like that. In the next, let's say 45 minutes-ish, I will talk about some new or rather new methods to evaluate spectroscopic data from combustion. In order to not run out of time in the end, I would like to acknowledge some of the contributors to this work, especially Christina Eisen. She was the person who basically started doing chemometrics in my group almost 10-15 years ago. And she's still doing chemometrics in industry now. So it's a quite nice toolbox that Chemometrics provides in all kind of fields. She's now doing research in pharmacy. So Stella Corsetti, Claudia Rullich, Anja Lampe, Florian Zehnbauer, Malte Stoth, and Udo Fritsching, and Andrew Williamson. Most of them are current or former students of mine. And yeah, for this work, we got financial support from the German Research Foundation, including a grant inside a priority program, which is focusing on flame spray pyrolysis of nanoparticles, or for the production of nanoparticles. And also we have some nice input from the CPAT Center, that's the Center for Process Analytics and Control Technology, which is a kind of consortium of universities and companies. It's organized by the University of Strathclyde in Glasgow. And the academic and industrial partners, they work together to find new ways to do process analysis, including developing software for chemometrics. So in this talk, I would like to give a brief introduction by talking about spectroscopy and combustion. Then I will introduce a couple of chemometric approaches to do data evaluation. I will then look into a couple of example applications. So we use chemometrics to evaluate spectra from fuels. We look into sprays and spray flames. And we also look into laminar and turbulent flames. So I will just highlight some of these areas, and then I will finish with a conclusion. So I don't think I need to tell you that combustion is quite important in our everyday life, and although there are efforts in politics to kind of reduce combustion, I think it will be very important for the foreseeable future. And so there's still a need to do combustion research and to kind of find out ways to make it more efficient, to make less pollutants. And yeah, basically this is why we are still working in this field, right? And one of I don't want to call it a problem, it's more a challenge. So combustion itself is a very complicated process. But in addition to the process itself, you have in technical combustors, you have commercial fuels, and they are complicated mixtures. So you have a mixture of, it can be tens or even hundreds of individual components. And that also makes it even more difficult to investigate and understand combustion processes. So what we are looking at are analytical methods, which are in the best case non-intrusive, so they do not impact the system we are analyzing, and they should provide chemical information, thermochemical information, also flow information. And if you ask modelers what they would like to have for a comparison, they want to have it in 4D, so spatially resolved, temporally resolved. And of course, the method should provide multiple parameters at the same time. So this is the kind of wish list we are aiming at. And spectroscopy offers a nice toolbox to achieve that. So spectroscopy, as you all know, it's utilizing the kind of interaction between light and matter. So the polarization is basically the response, and then you have the electric field or multiple electric fields which are interacting with your material. In this case, it's a gas or it's a fuel, and we're looking at this response, and we try to extract information about the material. And it can be a quite simple method like the one here. So you have one laser and you have a simple kind of detection system, or it can be a complicated method where you have to overlap multiple laser pulses in order to generate the signal. In general, we can say typically we have a rather sophisticated method and we have a challenging environment, and this results typically in a quite complicated data set. So what we want is we need data analysis methods that can deal with large data sets, and we need data analysis for complicated data. And there are a number of approaches that we can make use of. The first one is that's basically what we use. In most cases, it's univariate data analysis. So we have a signature in our spectrum, and we try to correlate it with the kind of quantity we want to measure. So we do a calibration, and this is what you can see here. So in this case, it's an intensity ratio, and it's calibrated against the equivalence ratio. What you can also do, it's a bit more complicated, is spectral modeling. This can be a kind of empirical modeling or a physical modeling. So in a physical modeling, you would basically, from first principles, for example, you would try to calculate the spectrum, and then you compare it with what you have measured. And from a library with varying parameters, with varying conditions, you basically get the information you want. This is what is often done in Raman spectroscopy or car spectroscopy. You can also do a more empirical approach. So you have a complicated spectral signature, and you fit it with a least squares algorithm to a number of let's say individual Gaussian or Lorentzian or Fock profiles, and you try to interpret these kind of individual contributions to your signature in the end. And the third approach is chemometrics. And chemometrics, for those who have not heard about it, I basically printed out a definition that was mentioned or said in a recent book. So chemometrics is the chemical discipline that uses mathematical and statistical methods to design or select optimum procedures and experiments, and to provide maximum chemical information by analyzing the chemical data. So the approach is you have an experiment, you produce data, you throw them in your chemometric tool, and you hopefully get the information you want. normally you get much more information than you want, and so you get relevant and unrelevant information. And so it can be quite difficult to basically to interpret the data or the information you get from a chemometric analysis. So what I would like to do in this talk is to give you a kind of brief overview of some kind of state-of-the-art methods and how we use it to analyze spectra. So chemometrics provides a huge toolbox, and unlike, let's say, 15 or 20 years ago, most of these methods are now implemented in software packages like MATLAB. So it's not that difficult to apply these methods anymore. So 15 years ago, you needed to basically get into all the mathematics. You had to sit down, write the code yourself. Nowadays, most of these routine methods, at least, are implemented in the common software tools. So one of the most important ones is principal component analysis. I will explain that in a minute. The beauty of that one is it's an unsupervised tool. So you have your raw data, you throw it into your algorithm, and you get some information back. And I will, as I said, I will show you what kind of information that will be. And then we have supervised methods. That means you need some a priori knowledge of your data in order to kind of apply these methods. So a very important one is partial least squares regression, PLSR. Like other regression methods, you basically try to understand or find correlations between different observables and the variables you want to measure. So typically you need a quite large training data set in order to develop your regression model, and then you can use that to basically apply it to unknown data. Those are the two I will talk about later. Then there's a huge class of methods called support vector machines. This is also a type of regression analysis, but you project your data, which you cannot find a good correlation in your original dimension, and you project it into a higher dimensional space where you can find this correlation. So so this is a bit more complicated. And then even more complicated are artificial neural networks. And as I said, those are the two methods I will not talk about today, because 45 minutes is not enough to give a full introduction to them. So to start with principal component analysis, it's basically a sort of unsupervised reduction of dimensions. So if you have spectra recorded from, let's say, a fuel or from a combustion system, then you have a highly complicated data set, and it's a multidimensional data set. So you can vary different parameters, of course. You can record spectra from different positions in the flame, for example. And what you end up with is basically a cloud of data points. So this is just a kind of schematic one. I will show you later how it looks like when you do that with spectra. So for example, spectral intensity as a function of wavelength. So you have a cloud of data points, but there is a lot of information that is basically redundant. So what principal component analysis does is you look for kind of new coordinates, and in a way such that you look for a first coordinate, which is called the first principal component, which represents the largest variance of your data set. So, for example, in this case, along this Z1 axis, you have the widest spread of your data points. So this represents the largest variance of your data. And then in the next step, you look for a second axis, which is orthogonal to the first one, and you again look for the direction which covers or which represents the largest remaining variance, and so on. So you do that in as many dimensions as you want. So mathematically, you reduce your experimental data set to a so-called score matrix T and a loadings matrix P, or in this case, the transform of a loadings matrix. And as you can see, I mean, they are smaller together than the original data set. And by looking at the variance, which is represented by these principal components that I've described here, you can basically decide how many principal components you need to get a quite good representation of your full data set. So in most cases, if the data are not too complicated, it's perfectly fine to have 2 or 3 or 4 principal components in order to present basically the major part of the variance of the data set. So the score matrix is basically now the original data in this new coordinate system, in the new space, and it reflects the sample distribution. The loadings matrix, on the other hand, is the contribution of the original data to creating the component. So you basically get kind of weighting factors for each individual data component. And from that, we can get some information about the variable intercorrelation. And just to give you an idea of how that looks like in a kind of real world system, I've put up some data where we use this PCA analysis, not in a combustion system, because that's quite complicated, but in a system of edible oils. So in this case, we were looking at passion fruit oils. So we had different samples, 5 samples from different manufacturers of passion fruit oils. We also had a sunflower oil just as a kind of reference. And when you look at the spectra here, those are IR spectra, they look very similar. So with the naked eye, you can hardly see any kind of differences. When you now apply principal component analysis to this data, so we used, of course, not only one spectrum for each sample, we used multiple spectra. And then we ended up with a score plot like this. So the score plot shows us some clustering of data. So all the kind of data points that have similar signatures, they group together. And this is quite nice, especially if you have, let's say, multiple samples and you record spectra from them. It's always nice to figure out how they basically, how close they are in such a score plot. And what you can see here is, so all the individual oil samples, the spectra from them, they stick together, which is a good sign, which means the samples are quite homogeneous. And we have the sunflower oil, which is quite far away from all the other stuff. So it's clear that we can distinguish between sunflower oil and the passion fruit oils. And then we have a very close group of passion fruit oils here. So they are very, very similar. There's one a little bit, excuse me, there's one a little bit off. And the interesting thing was those three were produced with the same process, and that one was produced on a more kind of industrial scale. And so this was a kind of different mode of production. So we can even distinguish between different production modes of these oils here. And then there was one passion fruit oils that was quite far away from the others. And a deeper analysis showed that this was not a pure passion fruit oils. It was adulterated with, I think it was rapeseed oil. So even this one, even this kind of outlier or fraudulent sample could be identified by this very simple and unsupervised data evaluation. And in the third plot here, I plotted the principal components loadings as a function of the wave number. So you basically get these kind of weighting factors for each individual point in your spectrum. And you can see, for example, principal component 1, it's only this very strong signature here, just to give you an idea. So this principal component 1 represents the largest variance of signatures in this region. And then you have principal component 2 and 3. And you can easily see the signal to noise ratio is much, much worse. And they look look significantly different. So for example, there are sidebands and stuff like that, which can be identified and which are characteristic for representing this variance of the data set. The only thing we need to keep in mind when we do any kind of chemometrics, I mean, you can get quite nice information, but you always have to keep in mind that what you're doing is pure mathematics. So you sometimes find correlations, but it does not mean that there's a kind of a real kind of scientific reason for that. So it can be just by accident that something goes up and another variable goes up in the same direction, but there's no kind of real physical meaning in between them. So this is something we need to keep in mind. It's pure mathematics, but it can be really helpful. So the other method I would like to mention is the PLSR. It's basically a regression model between some input data and output data. And of course, since we want to measure a certain quantity with these kind of regression models, we need some a priori knowledge about the data we put in. So typically what we need is a calibration data set or training data set where we know, for example, the concentration values that we're interested in the end. Oops. And so with this calibration data, we develop a regression model. So we try to figure out what kind of signatures are representative for this, for example, species concentration. And with this regression model that is developed with known data, we go to unknown data and we apply it, and we can do some quantitative measurements. So there's another technique which is quite similar to this PLSR approach, which is PCR. It's called principal component regression. The main difference between them is in the principal component regression, you use the principal component analysis, and then you try to figure out, you basically do this regression approach with the principal components that are coming from the unsupervised PCA. In the PLSR approach, you also develop these kind of, I call them principal components, but they are actually not the principal components, because those principal components are already kind of modified towards the quantity that you want to measure. So you basically, you look for the component that best represents the variance of the data in a sense, or with a target to measure this specific quantity. So these methods are slightly different, and because the PLSR is more targeted, you will always get some, let's say, better results from a quantitative perspective than the PCR. Okay, so that was the kind of introduction to some of these methods, and I would like to show some applications now. And the first area we are quite active in is fuel analysis. Because fuels are quite complicated, especially when it comes to commercial fuels. For example, natural gas, you can find, I mean, natural gas itself is quite complicated, but nowadays it's also mixed with biogas. There can be a mixture of hydrogen, which makes it even more complicated. Then you can have gasoline, petrochemical gasoline, which is blended with bioethanol. You can find diesel and biodiesel mixtures. Nowadays, they also put in some ethanol or butanol. So it's getting quite messy. And on the other hand, the fuel composition has a major impact, as you know, on the combustion performance with respect to efficiency and pollutant emission. So we need methods to actually analyze fuels. And in the best case, you have an analysis, and you use this analysis to control the combustion process. This is what we are looking for. In our lab, we use mainly infrared spectroscopy and Raman spectroscopy to analyze fuels. And so IR is a conventional absorption technique, and Raman spectroscopy is a scattering technique. Both provide information about the vibrational structure of the molecules in your sample. And the good thing is you get information about all the molecules at the same time. That's also a drawback because that makes the spectra quite complicated and difficult to interpret. So I would like to show you some example data from a study that we've carried out some years ago, where we looked at gasoline ethanol blends. So we basically produced our own gasoline, where we knew the composition, and we blended it with ethanol in different ratios. And then we applied Raman and FTIR spectroscopy, and we applied a number of different approaches to evaluate the data. And since our data were quite clear, they had a high signal-to-noise ratio, what we also did was we artificially added white noise to our data in order to see, so this is shown here, in order to see how this noise basically impacts the data evaluation. So what you can see here is the set of IR data for the pure gasoline. That's the blue dashed line, up to the ethanol, and the black lines in between are basically the blends. And the same for the Raman data. And yeah, what you can easily see in the IR spectrum, that's the OH band of the ethanol. And it's quite prominent in the IR spectrum because the OH bond is highly polarized. So it absorbs light quite nicely in the infrared. And on the other hand, in the Raman spectrum, you can find a strong signature here. That's the CH band, because they have a high polarizability. And on the other hand, the OH band is quite low in the Raman spectrum. So those were the data sets, and we as I said, we added some different levels of noise, basically from no noise up to, I think, about 20% of noise. And just to give you an idea of how that looked like, this is the one with 10% added noise. So as I said, we applied a number of different approaches, and I would say the simplest approach is if you do a univariate analysis. I go back one slide. If you look at basically this spectral region, you can see there is this large OH band, which is only there when there is ethanol in the mixture. And there is the CH band, which is always there because also ethanol has some CH groups. So you can easily calibrate basically the integrated intensity of this OH band, normalize it with the intensity, the integrated intensity with the CH band, and calibrate that as a function of your ethanol mass fraction or composition measure that you want to have. So we did that in different ways. We looked at the kind of full CH range. In some cases, we did only a kind of limited range here, because as you can see here, there are signatures which are dominant in the ethanol, and there are other signatures that are dominant in the gasoline. So in order to get a better distinction, we tried to kind of do this limited CH range calibration. And those are basically the calibration curves. So for IR, when you have the full range, you have this blue line here. And if you do the limited range, you get a better sensitivity. So it goes up here. But of course, since you have a limited range, noise might play a worse role here. And the same is true for the Raman results. With a full range, we're somewhere here. And then with a limited range, we increase the kind of sensitivity of this curve. And in order to look at how these or how the different evaluation approaches have an impact, we looked at the residuals of the mass fraction, which means we looked at what is our method providing, our data evaluation method providing as a value for the ethanol mole fraction in comparison to the mole fraction that we set when we prepared the mixture. So we basically subtract those two in a leave one out cross validation. So you use basically the full data set, but one data point for calibration, and then you add this one data point as a kind of unknown mixture, and you do that for each individual composition, and those are the residuals that we get. And you can see that there are no sort of strong systematic deviations here, but you have some sort of residuals here. And so there's always a sort of small measurement error in this approach. So in the next step, we looked at the noise impact. So we changed the levels of noise, and we determined the coefficient of variation of the root mean square error, and plotted that as a function of the noise level, just to see basically what is the impact of the noise. And just looking at those numbers, this kind of deviation or kind of error measure goes up quite significantly, especially in the Raman data. If you add noise, that's basically because you have this low intensity of the OH signature. It's much better in IR, so we could conclude that using this univariate method, the IR provides more robust results, especially in the presence of significant noise. We also did a multivariate analysis for all these data. And so we used the principal component regression. We also used the PLSR and did the same sort of data analysis. And those are the results, again, of the leave one out cross-validation. And you can see there's also some scatter, but the levels here are slightly, or about 30% smaller than what we saw in the univariate approach. So with using this multivariate techniques, we can get a more accurate kind of prediction of our fuel composition, which is a good thing, of course. We did the same thing with a multivariate evaluation as a function of noise level. Again, you can see a kind of similar behavior. The more noise, the less accurate your method becomes. What we did here, in addition to using the kind of CH signature here, we also used the full spectrum. So we looked at all the data that is there. And then, especially for the IR, you can see that even at 20% noise level, which is quite noisy already, you only have you have a rather good performance in terms of error. So that also shows that those statistical approaches, of course, they benefit from large data. So if you have a full spectrum, it can find signatures which may be not there for the naked eye, but it will find them, and you can get a quite accurate measurement result there. So it enhances the performance quite a bit. In Raman, you can deal with small levels of noise, but then, of course, at some point, the error becomes larger again. Okay, so that was the part on fuel analysis. So I would briefly show one example, what we do with principal component analysis in spray flames. So spray flames, the ones we are looking at, are used to produce nanoparticles. So you have, let's say, a methane oxygen turbulent flame, and you inject a mixture, a liquid mixture of a metal precursor that can be, let's say, an iron nitrate, or can be even a metal organic component. And the idea is to kind of evaporate those metals and produce small nanoparticles. And we want to understand the role of the chemistry inside the liquid in the production of the particles that come out in the end. And what we do in these flames is, for example, Raman spectroscopy, because we want to measure the temperature. So temperature is, as in any kind of combustion system, is a crucial parameter. And we try to understand these parameters here. So in this case, we recorded Raman spectra at different heights of above the burner. And one of the problems, if you have these kind of highly turbulent let's call them partially premixed combustion environments, at small scale, you will always, or in many cases, find some sort of a mixture inside your measurement volume. So you have a mixture of hot and cold gas, for example, or in this case, you might even find some liquid there. And it's quite difficult to figure out or to kind of pre-select the data in a way that your results are not biased by this kind of spatial averaging effect. So those are the nitrogen bands, the vibrational nitrogen bands that appear in the Raman spectrum. And what we are interested in, that's shown here in this small inset, we want to evaluate the temperature from the ratio of the cold vibrational band and the first hot band. So we calibrate this ratio as a function of temperature and get the information there. So we looked at two different cases in this study. So in both cases, we have fractions of hot and cold gas. But in one case, we assume we have an overall constant temperature of 1000 Kelvin. But the fractions that contribute to this temperature change in temperature difference. So you start with two fractions at 1000 Kelvin and you end with two fractions that are in total 1400 Kelvin away from each other. And what you can see here is if you have this, or the larger this temperature difference becomes, the more biased are your temperature results. And in the second case, we assumed a kind of premixed case in a sense that you have cold gas and you have hot gas in your system. So 300 Kelvin and 2000 Kelvin. And if we do that, we see that if we have small fractions of cold gas, the results in the hot gas region are quite biased. While if you have cold gas and you add a little bit of hot gas, the bias is not that significant. It's a little bit, but it's not that significant. But this is something you cannot tell by looking at the spectrum. That's the main problem. So what we did was basically looking at the principal component analysis of these data, and we developed a method where we look at or we calibrate our system with unbiased data, and this gives a nice curve here. And when you have biased data points, they deviate more or less significantly from this original behavior. So the more they deviate from this original behavior, the more temperature difference and the more temperature bias you have. And the same is true for the case 2 as well. So again, from the unbiased data, you get a nice curve here. And the more significant the error is, the more deviation you have from this curve. So in the last part, I would like to show some results where we use these chemometric approaches to laser-induced breakdown spectra. I must say that is work that has started, I think, almost 20 years ago in Lund when I was there as a research fellow, and we continued it in my lab now in Bremen, and we spent a lot of time developing new LIBS methods and now developing new approaches to evaluate the data. So in LIBS, it's also quite common to do a univariate method for data analysis. So you calibrate an intensity ratio as a function of equivalence ratio, for example, or you can even get temperature information if you look at other measures. And what we try with Chemometrics is to find ways to get all the kind of information at the same time. So in this case, we looked at a dimethyl ether air flame. We recorded in total 8,400 spectra. So it's a quite large data set. They're all plotted here, and you can see it's quite a mess. And it's almost impossible to evaluate all these spectra by hand. I mean, it's just too much. So what we did was we applied principal component analysis to this data, and this is so-called Biplot. It's also quite messy. So the blue signatures here are the coefficients from the PCA, and the red dots are the scores. And what is plotted is basically component 1, component 2, and component 3. So the first 3 principal components. And we figured out later on that basically 2 components are already quite significant or quite sufficient to provide or to present the whole variance of the data set. So this is why I'm going to the component 1, component 2 plot here. And this is now a biplot in order to make it a bit less crowded. We used selected spectra from a calibration measurement where we basically varied the composition of the gas. And you can see here again the coefficients, they are in blue, and they basically line up here quite nicely. And if you look at the kind of spectra they represent, you will figure out that we have the pure air in this region over here, and we have pure DME over here. And this is basically a variation of the DME concentration. So the chemical information is there. The red dots are the scores, and they represent basically the signatures inside the spectra. So the dots here that you can see over here, they are from the nitrogen signature at around 500 nanometers. Those dots are from the laser signature. So we have some elastically scattered light here. Those dots are from the hydrogen signature at 656. And since we have a fuel with some oxygen inside, and we have air with oxygen, we have the oxygen signatures, they are grouping over here and make it a little bit more messy. But yeah, as I said, all the information that we want to have is already there. And to give you an impression of how these principal components look like in a kind of spectral representation. So this blue one is the first principal component. The kind of reddish one here is the second one. And then there's a yellow one giving the third principal component. And yeah, this is what basically comes out of the statistics approach. Yeah, with that, I think we are close to coming to an end. I hope I could give you a kind of introduction to chemometrics. And chemometric methods basically facilitate the evaluation of large and complicated data sets. They can provide chemical information from a rather pure mathematical and statistical analysis. But this is also something to keep in mind. If you find a correlation, it doesn't mean there's a kind of real physical meaning behind it. It's just a correlation. So we need to think about what those kind of information, those pieces of information that come out of the Chemometrics toolbox, what they actually mean. So this is our task as researchers. A very good thing is normally you don't need any data pre-processing. So if there is a sort of background in the data, if there are signatures that you don't want there, in most cases you don't have to remove them because you do statistics, and in the end they will not play a role here. And I have shown you two different individual approaches. The first one was principal component analysis. The good thing here is you get all the features and correlations in a single plot. It's an unsupervised method, so you don't need any kind of a priori knowledge of your system. You can use that for system characterization. You can use it for identification of outliers or bias data. So it's a very helpful method, and it's very quick. You can find a routine, for example, in MATLAB. So writing the code here there to apply such a method takes 5 minutes, basically, if you know what you're doing. Then I've shown you some progression models, PLSR and PCR. They are good for a quantitative prediction, but you need a calibration and basically a development of this regression model. And of course, the more calibration and training data you have, where you know what's going on, the better the results will be. So with that, I would like to thank you very much for your attention, and I'm happy to take any questions.
Great, thanks so much, Johannes. Great talk. It's now time for the question and answer session. We have a number of interesting questions from the audience. Let me just dive right in with a big picture problem from Jay Gore, who asks, how do we convince the global society outside of the combustion field that it is not possible to stop using combustion as a process?
That's a good point. I mean, to me, it looks a bit like the whole discussion we had, at least in Germany, with nuclear power some years ago. Some people suggested that it's as easy as basically you shut down a nuclear power plant and you can raise one windmill and everything's fine. And yeah, I guess it's especially for special applications like long distance flights. I don't see any kind of electric or whatever propulsion approach that will be efficient enough to do transatlantic or transpacific flights in the foreseeable future. I guess we will rely on gas turbines and things like that. So yeah, convincing society that it's necessary, I guess it's tough, because there's a lot of, let's say, very polarized information in all the media. I mean, you have those kind of, not sure if I should call them radicals, but those radical green people who believe we can get rid of all the combustion. But I guess you need a more differentiated view of that. It's yeah, it's probably, I mean, you will always have parts of the society you will never convince. It's yeah.
Great. Okay, let me move on to the next question from the audience. One is, a potential problem with unsupervised machine learning methods is data integrity. Is there any way to be sure that the raw data being used is reliable?
Yeah, I mean, that's one of the things I mentioned in the end of the talk. You need reliable training data set, and the more training data you have, the more reliable and robust your approach will be. It's even more important when you use things like artificial neural networks or support vector machines. So developing these methods and making sure they are working requires a lot of work because you need to record all these data, and you need to characterize these data with other methods. So you need to know, for example, to train the data, to train the machine that this is a spectrum that is characteristic of this chemical composition, this temperature, and so on. And if you don't have such, let's say, reliable input data, yeah, the results you get are probably quite useless. But you will get results. That's probably one of the problems. You will always get a result.
I imagine that could pose some real difficulties.
Yeah.
Okay, next question from the audience is, it is rather difficult to perform Raman spectroscopy in flows with particles or droplets. What sort of particle number density did you have in your spray flames that you were able to perform Raman spectroscopy in there?
I don't have a number of the particle density. I can say it's a nightmare. So, I mean, I started looking at sprays with Raman spectroscopy during my undergraduate studies about 20 years ago. And it was a nightmare then. And I swore that I will never touch a spray again. And when I finished, the first thing I did was writing an application to get money for spray investigations. So it's yeah, it is a nightmare, and there are, or a significant fraction of the data that you produce, basically you have to throw it away because there's a laser breakdown, or there's an obvious kind of bias from elastic light scattering and things like that. So it's, yeah, it's difficult.
Okay, great. Next question from the audience is, autoencoder-based methods are now used to get interesting latent space representations beyond PCA. Do you expect it to replace PCA, or do you expect PCA to still dominate for its robustness?
Yeah, I mean, the answer is almost given already. I think PCA is so simple and robust. Of course, if you want to get some additional information, it's nice to have more sophisticated tools. But in my lab, we often use it just as a kind of initial screening tool to figure out, are there outliers? Are there any kind of strange things going on? Because you don't, yeah, there's no kind of effort to write the code for MATLAB. It can deal with large amounts of data. So I showed you these 8,400 spectra. I rerun the code just a few hours ago, just to get a feeling how it is today. And so within less than a minute, you get the results.
Wow.
So it's yeah, and that was on a kind of ordinary laptop computer. It's not a supercomputer.
So certainly a powerful technique.
Yeah.
Okay, the last question from the audience in the chat window that I can see, unless any come up, is, are there data sets available for teaching purposes? I could imagine that could be useful to many in the field.
In the end, you probably need training data that is somehow related to your experiment. So I'm not sure how useful it is to use a training data set unless it's recorded with the same sort of equipment and settings. So so to my knowledge, there are no kind of databases available where you can just take, let's say, Raman spectra from methane air mixtures, and you use them as a training data set for your method. I guess for these kind of standard cases, it might be a good idea to develop something like that. Yeah, okay, maybe something to follow up.
Sure. Yeah, it might be something interesting to follow up on. So I think it's time to close out the webinar for this week. Thanks so much for giving a really interesting talk. And if people would like to turn on their cameras and just thank the speaker, we can close out for the day. Thanks.